Hey there. Um, I've got a quick little thing I wanted to show you. It's related to what we've been talking about. Um, just some interesting things that I've seen in the news recently, and it helps to pull together some of the things that we've talked about. And you can see then how each of these things that we've learned about in class, how we can put them together and to form new ways to treat things like cancer. And so it's very brief, just a little example thing. And so one of the examples I want to show you has to do with gene therapy. And this particular example has to do with treating a particular type of cancer, but gene therapy in general is a fast growing field and it's very important to medicine, and the more we're learning about DNA and the human genome, and the more we're able to come up with treatments like gene therapy. And so this is something that can be used in lots of different diseases. But this is a good example of a way to treat a particular type of cancer called lymphoblastic leukemia. And so the FDA recently approved this, and the trade name is Chimeria. Chimeria? I'm not sure how to pronounce that. Anyway, it's gene therapy for this type of leukemia, which is a cancer. And what this therapy does is you remove the patient's T cells. So you take, a lymph sample from the patient and you isolate their own T cells. You remember the T cells are those lymphocytes that mature in the thymus, and of course they're very important to the immune system. And the gene therapy part here is that we take a gene and insert that gene into the T cells. And so white blood cells have a nucleus and they have DNA. And so we have this gene that we purposely insert into the DNA of the T cell. And you remember, genes are parts of a chromosome that code for RNA, which has the instructions for a protein. The DNA makes the RNA, makes the protein. And so this gene also makes a specific protein. But what that protein does is it makes the T cell target the cancer cells. And so then we take those modified T cells, inject them back into the patient, and then the T cells go and kill those tumor cells. And you can understand why we take the T cells from the patient and modify those T cells directly. If you just took anybody's T cells and then you injected them into their body, of course, those T cells would be recognized as non-self. And so the immune system would attack those T cells and it'd be a mess. But by using the patient's own T cells, you kind of avoid that. And you'll recall that we talked about that there already are some cancer cells that do get attacked by the immune system. This is just expanding upon that. And it seems to be very effective, but it's got really severe side effects because you're messing with the immune system. And so it's like a lot of chemotherapy in that, where the treatment is pretty harsh, but it's better than not treating the disease. And so that new gene that we put into the T cell, it makes a protein that binds with a protein on the surface of certain leukemia cells. Remember that some tumors produce unique surface proteins, and that's how your immune system is able to recognize them and attack them. And in this example, by identifying the surface proteins of this particular type of cancer, then they were able to genetically engineer a protein that would bind to those surface proteins and then put that gene into the T cells. And so the T cells match up to the leukemia cells. And you remember there's something called like natural killer cells or killer T cells. And their job is to go find cells and kill them. Now, remember that we talked about things like macrophages that are white blood cells that go and find a pathogen and eat it, engulf it, and digest it. But other cells, like the natural killer cells, they can't eat another cell. You know, it's too big to eat. What they do is they go poke holes in it, and they go poke holes in the membrane. And again, you remember what we said about membranes. They're semi-permeable. An important characteristic of life is that you keep the internal environment of the cell different from the external environment, and that cell membrane plays a very important role in that. But if you poke a bunch of holes in that membrane, then that membrane can't control what goes in and goes out, and the cell dies. And so that's how some of these white blood cells kill other cells. And so what we're doing is we're using those white blood cells, those T cells, to go kill tumor cells. And how does it know what cells to kill? Because we put this gene in that allows it to bind to and match up to those tumor cells, but not other cells. And so this is, just an interesting theory. And this is a growth industry. This is where lots of medicine is going to be take place, and where medicine is going to grow as we learn more and more about DNA and the human genome, and lots of new therapies for lots of diseases are based upon this idea. Use the patient's own immune system to kill the cancer cells or to attack other cells, because your immune system is designed to do just that. And if you can make your immune system recognize the cells that need to be killed, well, then you can just turn it loose. Another interesting treatment that I found is using anthrax to kill tumors. And anthrax is a very potent toxin, and it's a scary thing that has the potential for to be used as bioterrorism. And it's a toxin produced by a particular bacterium, Bacillus anthracus. And so in this treatment, the anthrax toxin is used to kill blood vessels which are feeding a cancerous tumor. Remember what a tumor is. It's a group of cells that are dividing uncontrollably. But they're living cells, just like all your other cells. Just like all your other cells, they need nutrients, they need oxygen, they need water, they need to get rid of waste. So they need blood flow, just like all your other cells. And so with this particular treatment, what you're doing is you're trying to starve those cancer cells. Instead of attacking the cells themselves, you're attacking the blood vessels which feed the cancer cells. And so this can work on any kind of tumor, anywhere in the body. And the anthrax works, got a protein, it's a protein that binds to the proteins on the surface of the blood vessels of the tumor. So again, it's by having this that specifically recognize particular blood vessels and killing just those blood vessels, then you can starve the tumor without having too many serious side effects. So that's also pretty interesting. Now in this particular treatment, you need to suppress the patient's immune system, because this anthrax is a poison, and it's toxic, and your immune system can recognize the proteins associated with the anthrax and try to eliminate it. And of course, you don't want that in this case. And so you need to suppress the immune system, which is always, again, it's a risky thing, but it's better than not treating the tumor. So this is not something that's been approved for humans yet. It's being tested in mice right now, but it is a potential treatment down the road. Okay. One other little thing I'd like to talk about, which again kind of pulls in stuff we've learned and is very interesting to me, is using viruses as part of the treatment. And so we've talked about viruses quite a bit and what they do, and obviously viruses are something that your immune system tries to attack. But scientists use viruses in lots of gene therapy. So for example, in that first example I gave you, I said we take a gene and we insert it into the chromosomes of the patient's T cells. Well, how? How do you put a gene into a chromosome of another cell? Well, there's lots of ways of doing this, and in fact, it's a fairly commonplace thing now for people who work with this. We just move genes into chromosomes basically at will almost. Um, so there are lots of ways of doing this, but one very effective way is to use a virus. And if you recall how a virus works, you can see uh, how this can be used. And so again, we've talked about viruses. What is a virus? It's a piece of DNA or RNA wrapped inside a protein coat. And here's a different picture than I've shown you before, but just these are different kinds of viruses, and you can see the genetic material is inside a protein coat. Sometimes it's DNA, sometimes it's RNA. And you'll recall that the virus attaches to the surface of one of your cells or to the host cell. And once it attaches to the surface of the cell, it's able to inject its genetic material into the cell. And then ultimately, the virus DNA inserts itself into your chromosome somewhere. And once it does that, then it's able to use the cell's mechanisms to make copies of the DNA and to produce lots of virus protein. And then you take all that virus DNA that you've just created, repackage it into new protein coats. Now you've got lots of viruses inside that cell, and the cell bursts and releases all those viruses, and it continues. So we've talked about this, and here's just another very simple diagram showing this, and this is a retrovirus, and so it's sticking RNA in, but some of them are RNA, some of them are DNA, and so there's a lot more details that we're skipping over here. The important thing to recognize is that viruses are good at inserting their DNA into the host DNA. That's sort of like their big accomplishment, and this is how they function. And so what we do is we take advantage of that, and we use the viruses for genetic manipulation. And so because they're really good at inserting themselves into chromosomes, we can take a gene that's useful to us, the gene that we want to use for treatment, and we put it in virus DNA. The viruses are simple, and so it's easier to put this gene in the virus DNA, and then we just let the virus do its thing, because the virus is very good at injecting its DNA into the cell and putting its DNA into the chromosome and making copies of that gene. But this time, when we infect the cells with the virus, it inserts the gene that we want to. And while we're manipulating the virus, we can, kind of manipulate it in such a way so that it doesn't make you sick and things like that. The point being is that we take advantage of the virus's ability to put itself in the host DNA to have it carry the gene we want into the host DNA. And so this is very common in genetic engineering, using viruses to put a gene into the target organism. Okay, so that's it. It's just a few interesting things that I've seen, and I just wanted to mention upon. I got nothing more right now, so let me know if you got any questions, but have a great day, and we'll see you.